Hello, this is Medicuri, and this video is an introduction to simulocyte. So, simulocyte is an online tool where it's basically what I would hope to become Minecraft for cell scientists. Different scientists go online, they log in, and then they start building 3D models of the molecular interaction of a cell, and then they can see what each others are doing, they can check each other's models, and they can even collaborate to be the bigger models. So the idea is that you can put as open source whatever data and models you're creating, but you can also protect them and create, a set of parameters where you can only allow certain users to visualize your model, because you might have invested time doing biological experiments to find data, and then once you publish, then you make your model available as openly as possible. So it uses WebGL technology, so you can visualize huge networks with like tens of thousands of nodes. And in order to access that system, you just need to go to the following web address. This is bio2fhms.surrey.ac.uk, and then you'll be directed to that login screen. If you don't have a graphic card, it's not going to work, because you need you need a graphic card, because this is WebGL technology, and you can display these huge networks only if you got a graphic card in your system. So if you got a desktop or a laptop with a graphic card, you're good. You also need Google Chrome. That's where the code has been optimized for Google Chrome, and it doesn't matter which platform you're in, Windows, Linux. Google Chromium works perfect on Linux as well, and Mac works as well. So as long as you got Google and a graphic card, you can access the system. You sign up as a user, you give your email address, blah blah blah. I'm not sending any emails to anybody. It's just that if you lose your password, you can click on some options to be resend a new password on your email. Alright, so I'm gonna log in. That's me, and it's still very new as a system, so it's not exactly beta, but it's freshly beta-ified, I might say. So the first screen you come upon is your user name, but we can consult the list of members, and when you look at that screen, you see who else is a member in this system, and who else you can see their email address, and you can see what models they have built, metabolic networks or models of the different chemical states of the cell, and so on. So it's a good thing, because you could check who's working on what, and you could email them if they have not given you access, so that you can work with them on something. So yes, there's also this little 3D visualization where it appears a bit like a cell kind of galaxy of users, and dynamically it will be populated if you've got more and more users in the system. And when you put your mouse on it, it tells you the name of the user. It just looks shiny, and I like it. Alright, so let's have a look on very superficially some of the features of the system. You can create by uploading SBML files. You can create metabolic networks, and this is just a 3D view of what the metabolic networks are from a quantity point of view. The size is logarithmic, so the number of nodes multiplied by the number of links and add the logarithm give you the size of this rectangle, and that just showed you kind of a sense of scale and how big these networks are without making them strikingly bigger or smaller. So yes, we got 3 metabolic network here which have been created as examples. Recon 2, which is based on the Recon X file, which compiles a huge number of metabolites and reactions to create a huge giant file showing all kind of metabolic reactions in the human cell. Then there is another example describing a subset of Reconex, which is the hepatocyte, which is the liver cell. And then there's another one, which is E. coli, which is the most studied cell probably in biology. So yes, you can see there's these three different things, and just to say the general control in this system are always the same. You use a mouse, you left click to rotate, right click to pan, and use the mouse wheel to zoom out or zoom in. So if I want to look at the detail of, let's say, Recon 2, I'll click on show. Then it's going to take a little while to show the network, because it's a massive network. It's several megabytes big as a file, but depends on your internet connection. Mine is not particularly fast, but it's still, visible, as you can see. So normally it shows at 60 frames per second, but as I'm recording a movie, it's only 30 frames per second at the moment. So this is a big, huge network in one big cell blob, which represent Recon to the different metabolic reactions inside the cell. If I make the lines more transparent or even invisible, I can start describing what's going on. So the little orange dot, which are actually round dots, represent metabolites, so things used by reactions or produced by reaction inside the cell. And the other dots in the bigger cone at the base represent reactions. So you could see red dots moving from each reaction to each metabolite, and that represent when a reaction used certain metabolite, then the dots just travel cyclically between. It's just a good way to locate which reaction are more important, because you can see that the dots converge in circular patterns towards certain metabolites or certain reaction, and that just means that some metabolites are used massively by lots of reactions and so on. You notice for the layout itself, it has this kind of flower pattern, because reactions can belong to different subsystems, and each different chunk of a different color represents a different subsystem. So if I put the mouse here, this is transport extracellular for that group, and this little other chunk here is transport mitochondrial. So it's a good way to separate reaction by their type. Okay, let me put the line back so we can x-ray this big complex network. You can see that some reactions are linked to a massive number of metabolites. Like this one, if I click on it, the name of this reaction appear, what gene expressing that thing, and the number of metabolites is linked to. Also, if I put the mouse on it, you notice that sometimes when I've got the data, which part of the KEGG metabolic pathway map are lit appear. So you can see this reaction lit this particular part of the KEGG metabolic pathway map, and that might be helpful, but it's just a partial data, because sometimes the file that you use to upload data to create this metabolic network don't contain this data. But when it has it, the system tries as best as it can to locate things on the map KEGG metabolic pathway data thing. Okay, so I wanted to also say that there is a system of tables where you can search information. So let's say you want to look for something that contain the word ATP, and you click enter, and then you select that thing, and it locates for you where it is. And it's also possible to mute certain things. If you got, for example, that thing here, which is quite massive, links to so many different things, you want to perhaps mute it because it's ATPase, and perhaps ATPase is just too many links in your network, and you don't need it because you know it's used in lots of things. So you're gonna locate it in your table by number. The number here was 10305, so 10305. You search for it, and it's there, and you can I'm gonna zoom it to show that you can actually mute that by clicking here on the checkbox. So up, muted, on, muted, on, muted, on. So you can mute lots of different nodes, and you have as well informations about the different types of reaction by subsystems, and you also have a hive plot. I'm not going to get into detail. I'll do other videos later to explain things in greater detail. Just also to show that there is a layout editor where people can create new 3D layout of the cell, and then once it's created they can save it, and other people, if they are given permission, can access these new ways to visualize the cell in 3D. So I've just shown you one big blob, but it's possible to, for example, download a layout that I made earlier, in which you can visualize the whole thing by compartments. And then you can see that there's 8 compartments, and the one at the bottom is the nucleus, the one in the middle is cytoplasm, the extracellular spaces at the top, and the organelles are satellites like around the cytoplasm. And just to say that you can see that some portions of the cell have very little link to each other. And you can of course zoom in and start looking at things in greater detail. So this is just one aspect of the system. People can build these models, they can look at them, and there's also the fact that we don't look just at metabolic networks, but also models, like you know, the way the cell reacts, what are the different chemical concentrations, and you know, for example, here there's lots of ways to build models of molecular interaction of the cell. Here we use quasi-steady state Petri net model. It's just one thing. I won't get into much detail about it at the moment. But here you can see there's 3 models available. The size of each one of these models reflect the number of nodes in these models. And there is on the left a little adjacency matrix, which is kind of giving you the signature of what this model connection to metabolic network is. For example, in here the columns represent the nodes in the model, and the rows represent the nodes in the metabolic network, and every time a node from a model is connected to a node from a metabolic network, you got a green rectangle. So when you see a line of green rectangle like that, a vertical line, that means that one node in your model is connected to a lot of metabolic network nodes. So one node regulates lots of metabolic functions. And so different models have different signatures, and it's a nice way to check the general look and feel of your model. Okay, well, we can look at the detail of a model. So I'll look at, for example, the old hepatocyte test sample model, and the liver cell model then. So it's gonna take a little while to download, but that's fine because it contains quite a lot of data. So you can see here in blue, this is your metabolic network. In green, this is your model, and the orange lines or yellow line are the connection between nodes in your model and nodes in the metabolic network. And it's also possible with the table to check what is where. You can edit the table of your models with the different different places and transitions, and there's lots of tables that shows you different things. And you can also as well, if you download the flux balance analysis, which is describing the rates of reactions and upper bound, lower bounds in your cell. If you download this data and you got the table, then for each reaction you can give it some kind of classification of subsystem. And when you do that, then I'm going to show you now, it's possible to visualize a very nice hive plot of the whole model, which actually make a surprising amount of sense. Okay, 1 second, I just need to find in my many files, where is this? And yes, so here I got an Excel file with a list of reaction and the different subsystem names they belong to. So I copy, I can paste that inside my little spreadsheet here, and what happens is then it associates automatically things from my data, and it shows my model as a giant hive plot where every axis represents a certain type of reaction, enzymatic on this axis, transport on this other axis, and so on. And you can see some nodes have a lot of roles, have a major importance with other bits of the network, and it's independent from network layout. It's really a purely functional network. Alright, so this is just a view of your model. It's possible to visualize different layout in 3D of your model as well. I'm not going to get into detail, but there's a lot of functions where you can visualize things completely differently. And then you can also make experiments. So once you've built a model, you can just show, you know, when you start your cell with certain initial condition, you can show what happens to the cell throughout your virtual cell in your simulation. So you enter certain parameters on how the cell starts and what the cell is trying to do, like make biomass, for example, and then the system will calculate what happens and then gives you back results once these results are computed. So it takes a while to download. I've got a fairly big experiment that I'm just downloading now, and just say that the general spirit of the thing is you log in, you build things, you see what other people have built, you share information, and then, you know, it's a bit like Minecraft, but for scientists. So yes, we got this massive model, and here you can see that the number of time, for example, some reactions are triggered or some genes are expressed, and you can see some things are expressed much more than others. For example, that thing here is never expressed or nearly not, and that thing is expressed massively during your simulation time. And also you can visualize different products or concentration throughout time. For example, if I add that to the pool of graphs, I can start seeing, you know, when something, that gene, when is it expressed, or when is that inhibitor triggered, and so on. And you know, you have here throughout time the different values, but also there is a horizon graph representation of this, which is useful for when you've got lots of things to compare to find cause and effect. Something which is used a lot in financial graphs. Okay, I can show a little nice neat feature, which is when you copy paste for each reaction what subsystems they belong to. It's now possible throughout the experimental result to see your hive plot, but also not just that, you can see the difference in concentration throughout time. For example, the blue line shows that it's increasing and the red that it's decreasing, and then you can show that the evolution of the relationship in your network throughout time when I displace the time value. Or it's also possible to just plot it differently, where the position on the hive plot represent flux values. So you can see that some things are going up and some things are going down throughout time, and so on. And I'm gonna pretty much stop there, because this is just a very fast overview. There's other types of experiment, but that's pretty much it for the moment. I hope you found it interesting. The code is available open source in Git Hub at simulocyte. So please have a look. If you want to contribute, don't hesitate, contribute. I'm very happy to have other people helping. And yes, thank you very much for listening. Bye-bye.